Hi everyone, and welcome to our video on osmoregulation. What we're going to be doing in this particular video is carrying on looking to see the function of our kidneys with the focus on specification reference 5.1.2d, the control of the water potential of the blood. First thing that we need to understand then is what on earth we actually mean by this phrase osmoregulation. Now, if we actually look at the word osmoregulation, we break it down into those two parts, then regulation is quite simply where we are talking about control. And osmo, hopefully, is familiar to us because we've heard of other words with osmo in it, such as osmosis, and therefore this is all to do with water. So basically what we're doing in our osmoregulation is we are controlling the water potential of the body. And in order to do this, we've got to control two aspects, water itself, and in order to control the water, we are going to also see some changes in those salts. The very basics of water is looking at how we gain it and how we lose it from the body. Now, in terms of how water gets into the body, through food, through drink, and obviously it will be produced as a product of certain metabolic processes, and it will be lost through those general excretory products. So we've got feces, urine, sweat, and of course every time you exhale, a small amount of water vapor is also lost. Obviously what we have to do here is ensure that we have the right amount of water within the blood at all times. If we don't have that control of the water potential of our blood, then what's going to happen is we're going to see those effects on the cells that we talked about back in module 2 where we were looking at osmosis. If too much water enters an animal cell, then it's going to make that cell undergo lysis. So basically, if we've got this very high water potential in the blood, that's going to lead to water moving into those cells, and therefore lysis could occur, i.e. the bursting of those cells. Not good news. If, however, we've got a very low water potential in our blood, then what we're going to find is that's going to lead to water being drawn out of those animal cells, like our red blood cells, for example, and therefore those cells are going to become crenated. And crenation, remember, is where we get that crinkling around the outside, and it affects obviously the effectiveness of those cells and their actions. In order to actually control the water potential of our blood then, what we're going to see is changes within the kidneys. And it's not just the overall kidney we're going to change, we're zooming in on those nephrons and focused on the collecting ducts. So what's going to be happening is we are going to alter the permeability of the collecting ducts. And remember, permeability is just how much water is able to pass through that particular plasma membrane. Now, what we're able to do, based on obviously whether the water potential in the blood is too high or too low, is we can then alter that permeability, making it either more or less permeable based on those current conditions. So, if we want to conserve less water, i.e. we have a surplus of water in our body, therefore a much higher water potential in our blood than we would like, what we are going to do is make the walls of the collecting duct less permeable. If the walls of that collecting duct are less permeable, that means less water is going to be reabsorbed into the bloodstream. We then have the inverse. If we want to conserve more water because the water potential of the blood is too low, then the walls of that collecting duct will become more permeable, therefore more water will leave the collecting ducts, travel into the capillaries and be reabsorbed into the blood there. So in terms of the actual mechanism, what we're going to see is a bit of an overlap with some other bits that we look at in module 5. And the first thing that we see is the hypothalamus. So this links into the work we've done on the brain and the different regions and what they actually control. So the hypothalamus is that region that's very much involved in that process of homeostasis in organisms. And in this case, we're obviously looking at the control of the water potential of our blood. So we're trying to keep that within a narrow acceptable range of values. So The hypothalamus then contains its receptors, and they're called osmoreceptors. If we break that word down into the two, receptor, obviously those cells that detect a stimulus of some form, in this case, changing water potential, and osmo, as we've already established, is going to be relating to water potential itself. So osmoreceptors, cells that detect a change in the water potential. Now, what's going to happen is, if the water potential of our blood is very negative, then that means that it is going to be more negative than the water potential inside those osmoreceptors. That means that water is going to therefore move out of the osmoreceptors into the blood, and as a result of this, those osmoreceptors will shrink. What we then see is the actual structure that we have up in that hypothalamus and the posterior pituitary region is that we've got these neurosecretory neurons that are extending their axons from the hypothalamus into the posterior pituitary, and they're going to terminate near the blood capillaries themselves. So that when we get these osmoreceptors losing water and shrinking, it's then going to send that impulse down the axon, and the end result is we're going to end up releasing molecules of ADH, antidiuretic hormone, through that process of exocytosis of those vesicles that will contain it. That ADH is obviously going to enter the bloodstream, and then it will travel through the blood until it reaches its target cells, i.e. the cells that have the receptors for ADH, and they are located in the collecting duct. So when that ADH actually binds to the receptors on the collecting duct cells, we then trigger a series of these enzyme-controlled reactions inside the cells themselves. And the end result of all of this is that we've got more vesicles within those cells of our collecting duct that contain these things called aquaporins. Now, aquaporins, if we think aqua is basically to do with water, and porins sounds like the word pore, like little holes. So basically these aquaporins are going to create little pores for water. They're going to basically act like a little hole to allow water to move. So ADH binds to those receptors. We then have our vesicles containing the aquaporins that will fuse with that cell surface membrane of the collecting ducts. And as a result, we're embedding those channel proteins, the aquaporins, into the cell surface membrane, and therefore water is able to cross those membranes really quite quickly. You can see I've given you a number here. You don't need to memorize it. 3 billion water molecules per second. So an incredibly effective way of shifting water from one side to the other side of the membrane. So what we're seeing here then is, because we've had that ADH lead to this vesicle being released, the aquaporins fusing with the cell surface membrane, we've got a more permeable collecting duct wall. As a result of that, more water is going to move from the collecting ducts into the blood capillaries and therefore be reabsorbed into the blood. If we've got a situation where the water potential of our blood is higher than the set point that it should be at, our osmoreceptors are not going to be stimulated, because remember, the whole way that they were stimulated was because of that lower water potential of the blood, therefore drawing water out of these cells. That's not happening here. The water potential is higher than we find within the actual osmoreceptors, therefore water is not going to leave, they will not be stimulated, and as a result, no ADH is going to be secreted. So in that scenario, we will no longer have the ADH binding to those receptors on our target cells in the kidneys. And instead, what we're going to see is a removal of those aquaporins. And all that happens to do that is the cell surface membrane of the collecting duct folds inwards around the aquaporins, forms a new vesicle, and basically removes them until they're required again. Now, because we've removed those aquaporins, there's less water that can then move through the actual collecting duct walls, and so less water is reabsorbed into the blood. As a result of that, more water remains in the filtrate, passes through the collecting duct, through the pelvis, and then down your ureter to the bladder. So, got a little flow chart here that just summarises everything that we've just looked at. And one of the things that you can actually do to help you learn this is basically print off or create yourself a version of this and then cover up each of those key parts on there. You know, post-it notes or just a little flap of paperwork, absolutely fine. Just stick anything on it. If you've got little scraps of card that you can put on there, anything works really. And then what you're going to do is just test yourself on it. So you are just going to start off, say, okay, if we've got an increase in water potential in the blood, next thing that happens is, and you just work your way around. And as soon as you can get all the way around this without any errors, you've got that first bit learnt. That doesn't mean it's locked away permanently, because we will have to revisit it a bit later, because as the brain works, hopefully we all know, we will forget things as time goes on. Therefore, revisit it a few days later and the few days after that, and the more you do that, then the more you're going to lock away in your long-term memory. So use that to help you in learning the process, but remember, osmoreceptors in the hypothalamus are the bits detecting the change. Then we're either releasing more or less ADH. As a result of that ADH getting to our kidneys, the collecting duct is going to become more or less permeable, and therefore that affects the amount of water reabsorbed. As always, I hope you found this video useful. Don't forget to subscribe to the channel so you can see when I next upload a video, and of course head on over to the website where you can find an array of other resources to help you in your A-level biology studies.